Organization of gene and chromosome. In molecular term, a gene commonly is defined as the entire nucleic acid sequence that is necessary for the synthesis of a functional polypeptide. According to this definition, a gene includes more than the nucleotide encoded the amino acid sequences of protein, referred to as the coding region. A gene also included all the DNA sequences required for the synthesis of RNA transcript. A typical prokaryotic gene. The figure B, a typical eukaryotic gene. A, the gene structure. Figure gene structure. The prokaryotic gene usually contains uninterrupted coding sequences. A, whereas the coding sequence of eukaryotic gene are commonly interpreted by non-coding sequences. B. Thus, a gene is the entire DNA sequences required for the synthesis of functional protein or RNA molecules. In addition to the coding region exons, a gene includes transcription control and regions, sometimes interons. Although many genes encode proteins, some encode tRNAs and rRNAs and other type of RNA. Most bacterial genes has no interons, whereas most genes of multicellular organisms do. The interons in human genes encoding average size protein are often much longer than the exons. Many bacterial proteins with the related functions are encoded by con- contiguous gene regulated by the single transcription control region. This type of gene cluster called as and is transcribed into a single operon polycystronic mRNA, which is translated to yield several different proteins. Most eukaryotic transcriptions units are transcribed into monocystronic mRNAs, each of which is translated into a single protein. The primary transcript producer from a single simple eukaryotic transcription unit is proceeded into a single type of mRNA. The primary transcript produced from a complex eukaryotic transcription unit can be proceeded into two or more different mRNAs depending on the choice splice sites and or poly adenylation site. Organization of gene and a non-coding DNA. The genome of prokaryotes and lower eukaryotes contains few non-functional sequences, whereas the vertebrate genome contains many sequences that do not code for RNAs or have any structural or regulatory functions. Only about 5% of genome DNA in a human encodes protein or functional RNA. The deficiency of consisting consistent relationship between the amount of DNA in the haploid chromosomes of an animal or plant and its phylogenetic complexity is called the C-value paradox. Human genome. This term is used to describe the different types of sequences that together makes up the DNA in the human cells. The human genome is 3.4 billion base pair in length. Only about 25% of DNA is transcribed into RNA, and less than 2% encodes protein. The active genes are often separated by vast deserts of non-coding DNA, much of which consists of repeated sequences derived from a transposable elements. The average characteristics of average characteristics of genes in human genome. Characteristics and average number of exons 8.8 average. Size of internal exons 145 base pair average. Size of interons 3365 base pair average. Size of 5 prime untranslated region 300 base pair. The size of 3 prime untranslated region 770 base pair. Size of coding region 1340 base pair. The total length of gene average 27,000 base pair. The DNA in the human genome is about 3 billion base pair long and is estimated to contain 30,000 genes. The DNA is arranged as a set of 23 chromosomes, each of which is a single double-stranded DNA molecule, 55 to 250 million base per long. The gene and gene-related sequences accounts for about 25% of DNA. The remainder is called a extragenetic DNA and has no known functions. Sequences in human genome. The human genome 3 billion base pair. The gene and gene related is divided into two types: gene and gene related sequence 20 to 30 percentages, and it is divided into two types: non-coding sequences approximately 90 percentages, and coding sequences approximately 10 percentages. Non-coding sequences approximately 90 percentages divided into intron, promoter, a leader or trainer, pseudo genes, and gene fragments. The coding and the human genome 3 billion base pairs divided into extragenic DNA 90 to 70 to 80 percentages, and it is divided into moderately highly repetitive 20 to 30 percentage, and unique low copy number 70 to 80 percentages. The moderately is divided into dispersed repeated 40 percentage, and clustered repeated 60 percentage G, capital G, and the dispersed repeated 40% divided into SINEs and lines, and a cluster repeated 60% G is divided into satellite DNA, mini satellite DNA, and a micro satellite DNA. This is a sequence of human genome. Gene and gene related sequences 20 to 30 percentage gene. The coding information in a gene is present as a series of sequence of DNA sequence called exons, separated from each other by intervening non-coding sequences called interons. The gene vary greatly in size and with respect to the number and size of the interons. Some genes, such as the histone H4 genes, are just a few hundred base pair long. Others, such as factor A8 gene, or several hundred base pair kilo base pairs in length and contains many large interons, such that the actual coding sequence accounts for just a few percentage of the total gene sequence. The additional sequences or presences which are associated with genes, the leader and the tailor sequences, occurs at 5% and 5 prime and 3 prime end of each of gene which are transcribed but not translated. The promoter may extend up to about 1 kilo base pair upstream, but other regulatory sequences that impacts transcription may occur at site much further away. 3.1 Gene families. Some genes exist as a few copies with identical or related sequences that can be grouped into families. The gene families may be organized in a few ways. 1. All the genes in the family occurs at this chromosomal locus. An example of this is the growth hormone gene families, whose 5 members are clustered on chromosome 17. 2. The gene belonging to the family may occur at a different loci. For example, the 5 members of the aldolase gene family are on different chromosomes. 3. The gene of a family may exist as a series of cluster of different chromosomes. An example is the homobox gene, which occurs as four each cluster on separate chromosomes, each containing about 10 individual genes. Pseudogenes. These are diverged member of a gene family that have acquired one or more inactivating mutation, so that they are no longer able to function and do not produce biological active proteins. The pseudogene is a simple a mutated version of a parent gene. The processing pseudogene, the deficiency interons, and because they do not have a promoter, they are not transcribed and do not result in protein production. It is it is not clear how proceed pseudogene arose in the human genome, but they may have been derived by reverse transcription of mRNA into double-stranded DNA, which was then inserted into the genome. Another group of inactive gene or gene fragments which deficiency the 5' or 3' regions of a parent gene. These are though to have arisen by a deletion event or by recombination that split the parent gene. The extragenic DNA 70 to 80 percentage. This part of human genome is composed of sequences that exist in addition to the gene and gene-related sequences described above in the figure. The extragenic DNA is composed of sequences that are not part of gene exons and introns and not associated with the gene leader and trailer sequences, promoters, and distant regulatory elements, and not a pseudogene or gene fragments. Although the extragenic sequences accounts for most of the DNA in the human genome, 70 to 80 percentage. That they have no known functions. Most of the extragenic DNA sequences, 70 to 80 percentage, are unique or exist as a small number of copies. The remainder 20 to 30 percentage are moderately or highly that may be dispersed repeated sequences throughout the genome or lined up end on end as long tandem arrays. Dispersed repetitive sequences. The two type of dispersed repetitive sequences exist, known as a short and long interspersed nuclear elements, abbreviated in science and lines. Science, SINES. The best known examples of science is ALU element. These sequences are not identical, but they are similar enough to be classified as a family. They have an average of 250 base pairs and almost 1 million copies exist, which are very widely spread throughout the genome, occurring in most places, including the intron of some genes. Two lines. They are similar to signs but have longer sequences. A well-known example is the L1 line, which is 6,500 base pair long and it is present as 60,000 copies. The lines are a type of retro elements. Clustered repetitive sequence. The human genome contains a extensive region in which the repetitive sequences are arranged end on end as long tandem arrays. This is called as a satellite DNA and occurs in various forms, each with different repeated units because they are separated from the bulk of cellular DNA by equilibrium density gradient centrifugation. Satellite DNA. Satellite DNA was the first type to be identified in human genome and contains repeated unit of up to 200 base pair arranged in cluster between 100 and 5000 kilo base pairs. Mini satellite DNA. The mini satellite DNA occurs in small cluster up to 20 kilo base pairs in length and has a repeated unit up to 25 base pairs. The length of a simple sequence tandem array is quite variable among individuals in a species, probably because of unequal crossing over during the meiosis. The microsatellite DNA. Microsatellite DNA occurs in short clusters, usually less than 150 base pair, and has a repeat unit which is usually 4 or less. Microsatellite DNA is very common. A variable number of tandem repeats. The length of microsatellite DNA sequences, such as the CA repeats and mini satellite DNA, varies from person to person. At any time, location in the genome, the number of repeat units present in an individual microsatellite DNA sequence may vary by as many as 10 or more. As such, these sequences are known as variable number tandem repeats or VNTRs. The polymerase chain reactions PCR can use to analyze the variable number tandem repeats. Figure the variable number tandem repeats CA is repeated 4 to 16 times PCR using primer specific for flanking unique sequences produces amplified DNA for varying lengths. Nature of gene and its function. The gene concept was introduced by Sutton The study of Morgan, Bridges, and Muller elaborated it. The essential feature of a modern concept of gene or the following. 1. The gene determines the physical as well as physiological characteristics. These are transmitted from parents to the offspring generated of the after generation. Since the number of genes in each organism is very large in comparison to the number of chromosomes, several genes are in each chromosome. In man, about 40,000 genes are known to be located on for 23 pairs of chromosomes. 2. Each gene occupies a specific position in a specific chromosome. This position is known as a loci. The chromosomal abbreviation like a translocation inversion may bring relative changes in the specific position of genes, and this helps in origin of chromosomes with the new sequences of the genes and helps in the origin of new species. 3. A single gene may occur in several forms or in several functional states. The form the forms other than normal are known as alleles. Many genes have only two alleles. One of them is normal and the other one is mutant. 4. Some genes mutate more than once and have more than two alleles. Some genes mutate more than once and have more than two alleles. These are known as a multiple alleles. Whatever may be the number of alleles in a multiple series, only two of them are found in an individual because of the presence of two homologous chromosomes of each type. 5. The gene may undergo sudden changes in expression due to changes in composition. The changed gene is known as a mutant gene and the phenomenon is known as a mutation. 6. The gene duplicate themselves very accurately. The phenomenon is known as a replication. Self-duplication of gene leads to chromosomal duplication. 7. Gene expresses themselves by producing enzymes which are proteins. It means each gene synthesizes a protein which acts as enzyme and brings about an appropriate changes. Molecular change of gene. Molecular structure of genes. Benzer has coined a new term. He denoted the relationship between the DNA molecules and the genetic phenomena. 1. Recon. It is the smallest unit of DNA capable of undergoing a crossing over and a recombination. A recon may be as small as one nucleotide pair in DNA. 2. Muton. It is the smallest unit of DNA which could undergo mutation. In its smallest expression, it represents a change in pair of nucleotides. Any changes in base pair of a triplet will modify the message carried by the codon. 3. Cystron. It is the unit of function. It is a gene in the real sense which consists of a few nucleotides and which is capable of synthesizing a polypeptide chain of genome. It means a cystron will consist of a nucleotides three times the number of total amino acids. A sequence of three nucleotide codes for one amino acids molecules present in the polypeptide chain for which it has the coded information. 4. Complon. It is a unit complementation. It has been used to replace cystone. Certain enzymes are formed of two or more polypeptide chains whose active groups are complementary to each other. 5. Operon. Operon is the combination of operator gene and sequences of structural gene which acts together as unit. Therefore, it is composed of several genes. The effect of operator gene may be additive or suppressive. 5. Replicon. It is the unit of replication. Several replicon constitute a chromosomes. Discontinuous gene, exons and introns. In 1977, it is discovered that the biological information carried by some gene is not continuous. It is split into several separate units or coding segments separated by the regions of non-coding DNA. Such genes are called discontinuous genes or split genes or mosaic genes. The sections containing biological information are called exons, and intervening non-coding sequences is called introns. The figure, the diagram of a hypothetical discontinuous gene showing the exons and introns. Interons. The introns are present in the gene of eukaryotes and their viruses, but these are absent in the gene of prokaryotes and their viruses. These are present in archaebacteria but are absent in mitochondrial genome. A single gene may contain no interons or may have as many as 52 as found in mammalian collagen gene. Gilbert hypothesis. It says that during evolution, the exons from different discontinuous genes can be shuffled, forming a new combinations of biological information. The shuffling may produce new functional proteins. The figure Gilbert hypothesis about the origin of new genes. Removal of interons from primary transcript. The transcription produces a faithful copies of a coding strands of gene. If the gene contains interons, its primary transcript will include copies of these. Therefore, interons must be removed and the exons regions of the transcript be reattached to one another before translation. This process is called splicing. Examining introns. The introns can be easily identified when nucleotide sequence of gene is compared with the nucleotide or amino acid sequences of gene product. The introns can also be detected by a method that depends on the ability of a gene to base pair to its RNA transcript and forms heteroduplex. A. If the gene does not contain introns, the mRNA will be continuous copy of DNA and an uninterrupted heteroduplex is formed. B. If the gene contains one or more introns, the heteroduplex will have a series of loops along its length. Self-splicing introns. The introns present in rRNA of certain protozoans splice and get folded up by intermolecular base pairing into a complex tertiary structure. Gene function. The genes acts mainly by producing enzymes which are involved in different steps of metabolism. The enzymes are proteins formed of polypeptide chains of amino acids. There are about 20 amino acids. Their arrangement determines different types of proteins. The sequence of amino acids in a polypeptide chain is determined by genes which contains the information in the form of the genetic code. The genetic code is the triplet sequence of a nucleotide base in the mRNA molecules which it has copied from the DNA molecules. The two theories has been put forward to explain the mechanism of gene action. 1. One gene, one enzyme theory. One gene, one enzyme theory was founded by geneticists Bedell and Tatum. While working one red mold, Neurospora crassa, for which they were awarded Nobel Prize in 1958. According to this theory, each gene in an organism controls the production of specific enzymes. The normal strain of Neurospora can grow in the minimal medium containing sucrose, salt, and one of the vitamin B biotin. It can synthesize all other complex substances of the culture medium. These biochemical synthetic reactions are brought about the specific enzymes. Note: the Neurospora presents a very suitable material for genetic studies because of the following reasons. A. The zygote is the only diploid stage. The vegetative phase is haploid. B. Zygote quickly undergoes meiosis. C. The product of meiosis, the ascospores in the ascus are arranged in a line strictly in the same sequence as they segregate at meiosis. D. It can grow in the test tube in a very simple culture medium containing the sucrose, some salts, and only one vitamin biotin. Such a culture medium is described as a minimal medium. The controlled experiment of Bedell and Tatum have shown that manufacture of biotin from simple substances of a minimal medium is completed in a few steps, and each step requires the presence of a specific enzymes, and each enzyme is produced by a separate gene. Let us presume that the synthesis of vitamin biotin 10 chemical reactions, and each one of them is governed by a specific enzymes. The S1 represents the raw material for the medium. S11 is the final product, thymine, and S2, S3, 2, S10 or the intermediate product. The enzyme involved during this process are repeated as E1, E2, E2, E10, and the gene synthesis these enzymes G1 to G10. The entire process of synthesis of thymine can be repeated. By adding the intermediate compounds, precursor of this medium, Bedell and Tantum were able to locate that steps in thymine synthesis, which was blocked in the mutant strain, resulting in the non-production of specific enzymes. On this basis, they presented one gene, one enzyme theory. One gene, one enzyme theory of Bedell and Tatum explains some of the human hereditary disease described by Garrod in 1908. US inborn error of metabolism. These are one, alkaptonuria, the urine of a person suffering from hereditary disease. Alkaptonuria turns black on exposure to air. This is due to the presence of substances alkaptone, homogenetic geneticic acid in their urine. Because in them the enzyme homogeneticic acid oxidase required in the conversion of homogeneticic acid into malacetoacetic acid is absent. These substances are intermediate in the metabolism of phenylalanine. The homogeneticic acid is passed out in the urine and later turns black on exposure to air. 1. Phenylalanine. 1. Phenylketonuria. 2. It forms tyrosine. And 3. It forms para-hydroxyphenylpyruvate. And 4. Alkaptonuria. It forms homogeneticic acid. And 5. It forms malacetoacetic acid. And 6. It forms fumaryl acetoacetic acid. And 7. It forms fumaric acid plus acetoacetic acid. This is the pathway of phenylalanine metabolism in man, showing the basic causes of disease alkaptonuria. The above observation indicates that in normal person the gene is present which controls the production of enzymes homogeneticic acid oxidase, and in alkaptonuria patients the gene at its locus fail to synthesize the necessary enzymes. 1. Phenylketonuria. This disease is caused due to the inability of metabolizing phenylalanine. As a result, the phenylalanine accumulates in the blood. This causes mental retardation, pale skin, and a tendency to epileptic seizure. 2. One gene, one polypeptide theory. According to this theory, one gene controls the synthesis of one polypeptide chain and not of the complete enzymes or protein molecules. This has been illustrated by sickle cell anemia, a disease of human being found especially in Negroes. The RBC in disease becomes sickle shaped in venous blood in lower concentration of the oxygen. This causes a rupture of cells and severe hemolytic anemia. The molecular basis of for the disease lies in the difference of only one amino acid molecules in hemoglobin of sickle cell anemic persons and the normal hemoglobin. Hemoglobin is a protein with a molecular weight of 64,000, containing about 600 amino acids. These amino acids are arranged in four polypeptide chains, the two identical alpha chains and two identical beta chains. The sickle shaped hemoglobin HB8 differs from the normal hemoglobin HB2 in the presence of valine in the place of glutamic acid in the polypeptide chains at number 6 of beta chain. The beta chain of hemoglobin A and beta chain of hemoglobin S. 1. Valine Valine it forms histidine. Histidine it forms leucine. Leucine it forms threonine. Threonine it forms proline. Proline it forms glutamic acid in case of beta chain of hemoglobin. In case of beta chain of hemoglobin, yes, it forms valine. And in both beta chain hemoglobin A and hemoglobin S, in stage 7 it forms it forms glutamic acid, and stage 8 it forms lysine. This is the figure of beta chain of normal hemoglobin and sickle shaped hemoglobin, showing differences in the arrangement of amino acids. The sickle cell anemia is therefore produced by a single change in 2 beta polypeptide chain out of the total 4 causes by a single mutation in one gene. It means that one gene controls the synthesis of one polypeptide chains and not the complete proteins. Genetic code. The genetic code is the set of rules by which information coded within the genetic materials, DNA or mRNA sequences, is translated into the protein by living cells. The genetic code consists of 64 triplets of nucleotides. These triplets are called codons. 3. The AUG codon is also known as interior codon because it's a signal that starts of translation and its codes for the incorporation of amino acids methionine MET into the growing polypeptide chain. 4. The UAAA, UGA, UAG codons or terminator codons. Note: all but two of the amino acids methionine and T or P can be encoded by from 2 to 6 different codons. Example: in humans, for example, alanine is encoded by GCC 4 times as often as by GCG. A. Adenine, U. Uracil, G. Guanine, C. Cytosine, and T. Thymine. Importance of important points. The triplet code, the three adjacent nitrogen bases consist a codon and specific one amino acids. 2. Commaless. These are no pause so that the genetic codes reads continuously. Any changes in nucleotide sequences will make the code read differently. 3. Number. The possible number of a triplet is 64 bracket 43, but amino acid involves are only 20. 4. Non-overlapping codon. The adjacent codon are independent with no base pair being member of two codons. 5. Non-ambiguous codons. One codon specifies only one amino acid and not any others. 6. Degeneracy of codons. The tryptophan is specified by only UGG and methionine by only AUG. All other amino acids are specified by 2 to 6 codons. Example: phenylalanine by 2 UUC and UUU, and arginine by 6 AGA, GGA, CGA, CGC, CGG, CGU. Cistron, the expressible part of genetic information contained in DNA, occurs in the linear segments called cistron. Polarity. Each cistron has a fixed polarity with a start point and an end point. Collinearity. The sequence of a codon of DNA mRNA corresponds to the sequence of amino acids in a polypeptide. Start signal. The first codon starting a polypeptide is AUG, methionine specific, but in prokaryote it can be GUG and even UUG. In all cases, they specify the methionine as the first amino acid, though GUG and UUG specific different amino acids inside the polypeptide chain. GUG valine and UUG leucine. Stop signal. The termination of a polypeptide synthesis occurs by one of the three nonsense or termination codons: UAA, Okiri, UAG, amber, and UGA, opal. Related codons. The amino acids with similar properties have usually related codons. Example: aromatic amino acids, tryptophan, UGG, phenylalanine, UUC, UUU, and tyrosine, UAC, and UAU. Degeneracy of code or Wobble hypothesis. Crick in 1966, 5' end of tRNA anticodon can wobble and get paired with even non-complementary bases of mRNA. Therefore, the first two bases of codon are more important in determining the amino acids, while the third one may differ without affecting the coding wobble loose pairing. Example: CCA, CCC, CCG, CCU, all specific amino acid proteins. It leads to degeneracy of genetic code. Nirengenberg and Metai deciphered the genetic code. RNA codon. In RNA, there are four types of nucleotides, which are adenine, uracil, guanine, cytosines. So, in RNA, following 64 codons are found. Note: in the table, the left-hand column shows the first nucleotide of the codon, and the fourth middle column shows the second nucleotide, and the last column shows the third nucleotide. DNA codon. In DNA, there are there are also found 64 codons, but there are adenine, thymine, guanine, cytosine nucleotides are present. They have some same codon, but thymine T replaces the uracil. Codon bias. All but two of amino acids, methionine, troponin, TRP, can be coded by from 2 to 6 different codons. For example, alanine is encoded by GCC 4 times as often by GCG. This probability affects a greater translation efficiency by the translation apparatus. Example: ribosomes for certain codons over their synonyms. Mitochondrial genes. When mitochondrial mRNA from animals or microorganisms, but not from plants, is placed in the test tube with the cytosolic protein synthesizing machinery, the amino acid enzymes, tRNA ribosomes, it fails to translate into a protein. The reason being the reason the reason being these mitochondria uses UGA to encode tryptophan (TRP) rather than as a chain terminator. When translated by the cytosolic machinery, the synthesis stops where TRP should have been inverted. In addition, most one animal mitochondria uses in animal mitochondria use AUA for methionine, not isoleucine, and 2. All vertebrate mitochondria uses AGA and AGG as chain terminators. The yeast mitochondria assigned all codons beginning with CU to threonine instead of leucine, which is still encoded by UUA and UUG as it is in cytosolic mRNA. Two cases have been found where an amino acid that is not one of the standard 20 is inserted by a tRNA into a growing polypeptide. 1. Selenocysteine. The amino acid is encoded by UGA. UGA is still used as a chain terminator, but the translation machinery can discriminate when UGA codon should be used for selenocysteine rather than a stop. This codon usage has been found in certain archaea, eubacteria, and animals. The human synthesizes 25 different protein containing selenium. 2. Pyrolysine. In several species of archaea and bacteria, this amino acid is encoded by UAG. How the translation machinery knows when it encounters UAG, whether to insert a tRNA with pyrolysine or to stop translation is not yet known. 3.2. Structure of chromatin and chromosome. The the nucleosome is a bead-like structure of a chromatin fiber. The nucleosome contains approximately 200 base pair of DNA organized by a octamer of small basic protein called histone. The nucleosome provides a first level of organization, giving a packing ratio of approximately 6. The second level of organization is the coiling of series of nucleosome into a helix array to constitute the fiber of diameter approximately 30 nanometer interface chromatin. In chromatin, this brings the packing ratio of DNA to approximately 40. The final packing ratio is determined by the third level of organization, the packing of 30 nanometer fiber. This gives an overall packing ratio of approximately 1000 in euchromatin. The figure shows organization of chromosome. Short region of double helix, short region of DNA double helix, 2 nanometer. 2. Beads on a string form of a chromatin of 11 nanometer. 3. 30 nanometer chromatin fiber of packed nucleosomes 30 nanometer. 4. The section of a chromosome in an extended form 300 nanometer. And 5. 700 nanometer the contains the section of chromosome. And 6. 1400 nanometer entire mitotic chromosomes. The nucleosome contains approximately 200 base pair of DNA associated with the histone octamer that consists of two copies of each of H2A, H2B, H3, and H4. These are known as a core histone. The histone H3 and H4 are among the most conserved proteins. The H2A, H2B shows up appreciable species specific variation in sequence. The histone H1 protein shows appreciable variation between the tissue and between the species and or absent from yeast. The length of the DNA per nucleosomes varies from the individual tissue in the range from 154 to 260 base pair. The figure shows the cleavage by micrococcal nucleus. It forms nucleosome of 200 base pair, and it is formed a trimmed nucleosome of 165 base pair, and it finally forms 146 base pair of core particles. The histone octamer has a kernel of a H32 to H42 tetramer associated with the two H2A and H2B dimer. Each histone is extensively interdigitated with its partner. All core histories have the structural motif of histone folds. The internal tail extends out of the nucleosomes. The histone octamer are not conserved during the replication, but H2A, H2B dimers and H32 and H42 tetramers are conserved. The RNA polymerase displaces the histone octamers during the transcription. The nucleosomes reform on DNA after the polymerase has passed, unless transcription is very intensive, such as in ribosomal DNA, when they may be displaced completely. It's a composition. Is a figure shows the nucleosome and the histone. The composition of a human genome. The human genome it is divided into nuclear genome 3,300 MB, approximately 80,000 genes, and two mitochondrial genomes 16.6 kilobase per 37 genes, and nuclear genome 3,300 MB, approximately 80,000 genes is divided into approximately 25 percentage and approximately 75 percentage. The approximately 25 percentage from gene and gene related sequence, 75 percentage from estrogenic DNA, and mitochondrial genome 16.6 kb 37 gene is divided into 2 ribosome on a gene, 22 T on a gene, 13 polypeptide encoding genes. The gene and gene related sequences, it forms unique or moderately repetitive, approximately 10 percentage, driven to 10 percentage, and approximately 90 percentage. Approximately 10 percentage is driven to coding DNA, and 90 percentage is driven into non-coding DNA. The estrogenic DNA is divided into approximately 60 percentage and approximately 40 percentage. 60 percentage is unique or low copy number, and approximately 40 percentage is moderate to highly repetitive. The non-coding DNA is divided into pseudogenes, gene fragments, and interons, untranslated sequences. The and in case of estrogenic DNA, 40% it forms moderate to highly repetitive. It is given to tandem repeat or clustered repeats, and interspersed repeats. This is the composition of a human genome. The table shows the genome size and the number of genes present in some organisms. Mycoplasma genitalium group Eubacteria, genome size 580 kilobase, and number of genes is 468. Escherichia coli group Eubacteria, the genome size 4639, and the number of genes is 4289. Helicobacter pylori, the group Eubacteria, genome is 1667, and the number of genes is 1590. Rickettsia and Provoski, Eubacteria, then genome size is 1111, and number of genome is 834. And in Methanococcus janaski group Orchibacteria, genome size is 1664, and number of genes is 1750. Saccharomyces cerevisiae, group budding yeast, the genome size is 12,069, and the number of genes is 6,300. Arabidopsis thaliana, group flowering plant, genome size 1 lakh 42,000, and number of genes is 26,000. Cyanorhabditis elegans, it's a group of worm. Genome size is 97,000, and number of genes is approximately 19,000. Drosophila melanogaster group fruit fly, genome size is 1,37,000, and number of genes is 14,000. Homo sapiens group humans, and the genome size is 3 crores 20 lakhs, and the number of genes is 30,000. Unique and repetitive DNA. The eukaryotic genome can be assessed by the reassociation of kinetic and denatured DNA. This technique was used extensively before large-scale DNA sequence. The reassociation kinetic identifies two general type of genomic sequences. One, non-repetitive unique DNA. It consists of sequences that are unique, that is, only one copy is haploid genome. Two, repetitive DNA. It consists of sequences that are present in more than one copies in each genome. Repetitive DNA is often divided into two general types. One, moderately repetitive DNA. It consists of relatively short sequences, 10 to 100 base pair in genome. The sequences are dispersed throughout the genome. It is also responsible for high degree of secondary structure, the formation of pre-mRNA, inverted repeats in the intron pairs to form a duplex region. 2. Highly repetitive DNA consists of a very short sequence, typically approximately 100 base pair in genome. These sequences are present many thousands of times in genome as a long tandem. Neither moderately repetitive nor highly repetitive DNA represents any protein. The length of non-repetitive DNA component tends to increase with overall genome size. The non-repetitive DNA content of a genome is synonymy with the relative complexity of organisms. E. coli has to 4.2 and 10 to the power of 6 base pair. C. elegans increases to 6.6 and 10 to the power of 7 base pairs. D. Drosophila melanogaster increases further approximately 10 to 10 to the power of 8 base pairs. And mammal increases another order of magnitude of approximately 22 into 10 to the power of 9 base pairs. The reassociation kinetics shows that mRNA is derived from non-repetitive DNA. The amount of non-repetitive DNA indicates the total DNA of coding potential. Many exons have related sequences. The exon sequences are conserved, but introns varies. Such exons evolved by duplication to give copies that initially are identical, but which then diverged in sequence during the evolution. The figure, the absolute content of non-repetitive DNA increases with genome size, but reaches a plaque at 2 into 10 to the power, to 2 into 10 to the power of 9 base pairs. The non-repetitive unique red, moderately repetitive blue, and highly repetitive green. Key points: the comparison of human and puffer fish, Fuga rubrips has 365 billion base pairs and about 30,000 genes. The human genome has one third fewer gene in 8 times the amount of DNA. The genome shows that many genes in two organisms are similar. The major difference between the two genomes is in repetitive DNA sequences, which makes up the 40% of human genome, but a much smaller portions in the puffer fish. 2. The prokaryotes contains only non-repetitive DNA. 3. The most of the DNA of lower eukaryotes is non-repetitive, approximately 20% or moderately repetitive components. 4. In animal cells, up to half of the DNA is occupied by moderately and highly repetitive components. 5. In animals, in plants and amphibians, the moderately and highly repetitive components may accounts for up to 40 accounts for up to 80% of the genome. The moderately repetitive DNA consists of transposons, short sequence of DNA approximately 1 kb that can move to new location in genome. Reassociation kinetics. When single-stranded DNA molecules are mixed, they may reassociate depending on the homology between the two type of strands. The facility of reassociation is determined by the degree of homology of the reactant, their concentration, and the time allowed for annealing. These three parameters are expressed by Scott curve. The redundant sequence palindromic DNAs can anneal rapidly because of their similarities. For unique sequences, it takes more time for the complementary sequence to anneal. The reassociation of equally DNA generally takes place between the COT values of about zero point 0.1 and 10, Ca 4.7 into 10 to the power of 6 base pair, mainly unique DNA, whereas for a large and complex genome of RYE containing about 80% of redundancy in the 7.9 into 10 to the power of 9 base pairs. The figure shows reassociation kinetics of two genome. 7.9 L 109 base pairs of RYE and 4.7 L 106 E. coli of different complexities. The repetitive sequence accounts for approximately 50% of the human genome. The repetitive sequence falls into five classes. 1. Transposons, either active or inactive, accounts for 40% of genome, 45% of genome. 2. Proceeded pseudogenes, approximately 0.1% of total DNA. Pseudogenes or dead end of evolution. 3. Simple sequences repeats accounts for approximately 3%. 4. Segmental duplication accounts for approximately 5%. 5. Tandem repeat sequence at centromeres and telomeres. 3.2. Heterochromatin versus euchromatin. The chromatin is divided into euchromatin and heterochromatin. The euchromatin comprises the majority of genome. The identified genome sequences represents approximately 90% of euchromatin. In addition to providing the information on the genetic content of the genome, the sequence also identifies the features that may be of structural importance. Euchromatin does not observe the common nuclear strains during the interface and is normally transcribed into the mRNA. In the human genome, the euchromatic fractions varies a great deal among the chromosomes. In chromosome 2, and almost 160 MB sequences is less than 60% of euchromatic, whereas a more than 30 MB sequences in chromosome 21 is almost entirely euchromatic. The figure: the gene occupies 25% of human genome, but only 1% of human genome consists of coding regions, protein coding sequences. The human haploid genome consisting of 22 autosomes plus the X or Y. The chromosome ranges in size from 45 to 279 MB of DNA, making a total genome content of 3286 MB, approximately 3.3 and 10 to the power of 9 base pair. The euchromatin comprises the majority of genome, approximately 2.9 and 10 to the power of 9 base pair. The identified genome sequences represents approximately 90% of euchromatin. Some regions of chromatin are very densely packed with the fibers, displaying a condition comparable to that of chromosomes at mitosis. This material is called heterochromatin. It is typically found at a centromere but occurs at other locations also. It often the various heterochromatic region aggregates into a densely staining chromocenter. This region is always heterochromatic, called constitutive heterochromatin. In addition, there is another sort of heterochromatin called facultative heterochromatin, in which the region of euchromatin are converted to a heterochromatic state. The common features of a constitutive heterochromatin are: it is permanently condensed to, it often consists of multiple repeats of few sequences of DNA that are not transcribed. 3. The density of gene in this region is very much reduced compared with the heterochromatin, and genes that are translocated into or near it are often inactivated. 4. It replicates late in S phase and has a reduced frequency of genetic recombination. The facultative heterochromatin behaves as a heterochromatin only under certain conditions, like in mammalian X chromosomes when present in more than single copy. The reduced acylation of histone protein, increased methylation of one histone proteins, and hypermethylation of cysteine base in DNA. These molecular changes causes the condensation of materials which is responsible for inactivity. Although active genes are contained within euchromatin, only a small minority of sequences in euchromatin are transcribed at any time. So location in euchromatin is necessary for gene expression, but is not enough for it. Satellite DNAs are found in regions of heterochromatin. The heterochromatin is a region of chromosomes that are permanently tightly coiled up and inert, in contrast with the euchromatin that repeats most of the genome. Heterochromatin is commonly found at centromeres, the kinetochores, the region controlling the chromosome movement. The centromeric location of satellite DNA suggests some structural functions in the chromosomes which relates to the process of chromosome segregation. Types of chromosome structure. The DNA molecules consist of two linear chains that are wrapped around one another, forming a double helix structure. These helical structures are further wound into the chromosome structure. The chromosome are divided into two parts with a constriction point in the middle, which is known as a centromere. The four types of chromosome in animal cells are classified by the position of centromeres. In the nucleus of each cell, the DNA molecules is packed into a thread-like structure called chromosomes. Each chromosome has a construction point called centromere, which divides the chromosome into two sections or arms. The short arm of the chromosomes labeled the Q arm, the long arm of the chromosomes labeled as P arm. The location of the centromere on each chromosome gives the chromosome its characteristic shape and can be used to help in describing the location of specific gene. The structure of chromosomes. The chromosomes are made of many chromatin thread, each containing DNA and proteins. 2. The chromosomes are made of 50% of protein, the structure, protection, turn gene on and off, and 50% of DNA, deoxyribonucleic acid. 3. The somatic cells containing 46 chromosomes, gametic cells containing 23 chromosomes, and 4. 44 of our chromosomes are autosomes, while 2 or 6 chromosomes. 5. Each chromosome contains many genes that acts as a blueprint or set of instructions for the cells. 5. 6. The electron microscope study later shows that there is no different membrane pellicle surrounding the chromosomes. The other structure present in the chromosomes includes chromatids, centromeres, secondary constructions, nucleolar organizers, telomeres, and satellite. Types of chromosomes based on number of centromeres. The number of centromeres present on the chromosomes helps in determining the type of chromosomes. These different chromosomes types based on the number of centromeres are as follows. 1. Acentric chromosomes. Acentric chromosomes are those that lack centromeres, that is, the centromere is totally absent in chromosomes. These chromosomes are observed due to the effects of chromosomes breaking process like irradiations. 2. Monocentric chromosomes. The monocentric chromosomes are those that contains a single centromere. This type of centromere is present in most of the organisms. The monocentric chromosomes can be called a acrocentric if the centromere is located at the end of the chromosomes. If the centromere is located towards the center, it is called metacentric chromosomes, and telocentric chromosomes are those in which the centromere is present at the chromosomes end. 3. Dicentric chromosomes. The dicentric chromosomes are those that have two centromeres that are present on its arm. These chromosomes are formed after two chromosomal segments with a centromere each or fused end to end. This causes them to lose their acentric fragments, leading to formation of dicentric chromosomes. 4. Polycentric chromosomes. The polycentric chromosomes are those that contains more than two centromeres. These chromosomes are very common in plants. For example, Ada's tongue fern that has 1262 chromosomes. They include the autosomes and sex chromosomes. Thus, in all humans contains 22 pairs of autosomes and 1 pair of sex chromosomes. This leads to the presence of 46 chromosomes present per cell. The chromosomes are very important for every organism, as the future of their progeny depends on the normal division and separation of chromosomes. Types and number of centromeres. Acentric number of centromeres is nil. Monocentric number of centromeres is 1. Dicentric number of centromeres is 2. Tricentric number of centromeres 3. Polycentric number of centromeres many. Holocentric diffuse. Here microtubules are attached along the length of chromosomes based on position of centromeres. Metacentric chromosomes. The metacentric chromosomes have the centromeres in the center such that both sections are of equal length. Human chromosomes 1 and 3 are metacentric. 2. Submetacentric chromosomes. The submetacentric chromosomes have the centromeres slightly offset from the center, leading to a slightly asymmetric in the length of two sections. The human chromosomes 4 through 12 are submetacentric. 3. Acrocentric chromosomes. The acrocentric chromosomes have a centromere which is severely offset from the center, leading to one very long and one very short section. The human chromosomes 13, 15, 21, and 22 are acrocentric. 4. Telocentric chromosomes. The telocentric chromosomes have the centromeres at the very end of chromosomes. The humans do not possess telocentric chromosomes, but they are found in other species such as mice. The metacentric, the position of the centromeres is center median shape V shape, details equal arms. Examples Tradiscantia amphibia. The type submetacentric, the position is near center submeridian, shape J or L shape, and two equal arms. An example human beings. Type acrocentric, position of centromere is near to one end subterminal, shape one arm very short and another long. Example grasshopper. Type telocentric, the position of centromere terminal, shape rod like, and example drosophila. The chromosomal anomaly and abnormality and abbreviations. When the chromosome structure is altered, this can take several forms. Deletion. The portion of chromosomes missed or deleted. Known disorders in human includes Wolf Hirschhorn syndromes, which is caused by partial deletion of short arm chromosome 4. Jacobson syndrome, also called the terminal 11q deletion disorder. To duplication, the portion of the chromosomes duplicated, resulting in extra genetic materials. Known human disorder includes Charcot Maria tooth disease type 1A, which may be caused by duplication of gene encoding the peripheral myelin protein 22, PMP22 on chromosome 11. Translocation, when a portion of one chromosome is transferred to another chromosome. There are two main types of translocation. In the reciprocal translocation, the segments from two different chromosomes have been exchanged. In Robert's Onion translocation, an entire chromosome has attached to one another at centromere. In humans, this only occurs with chromosome 13, 14, 15, 21, 22. Inversion, a portion of chromosome has broken off, turned upside down, and reattached. Therefore, the genetic material is inverted. Rings, a portion of the chromosome has broken off and formed a ring or circle. This can happen with or without loss of genetic materials. Isochromosomes, it is formed by a mirror image copy of chromosomes segment including the centromere. The gain or loss of DNA from the chromosomes can lead to a variety of genetic disorders. Humans example includes Cat, which is caused by the deletion of part of short arm of chromosome 5. Cri du cat means cry of the cat in French, and the condition was so named because affected babies may high pitched cries and sounds like those of cat. The affected individual have wide set eyes, a small head and jaws, moderate to severe mental health issues, and are very short. Down syndrome usually is caused by an extra copies of chromosome 21, trisomy 21. It is characterized. The characteristics includes decreased muscle tone, stockier build, asymmetric skull, slanting eye, and mild to moderate developmental disability. Edward syndrome, which is the second most common trisomy. The Down syndrome is the most common. It is a trisomy of chromosome 18. The symptoms includes motor retardation, developmental disability, and numerous congenital anomalies causing serious health problems. The 90% die in infancy. However, those that live past their first birthday usually are quite healthy. Thereafter, they are clenched hand and overlapping finger. IDIC 15, abbreviation for isodendritic 15 on chromosome 15, also called the following names due to the various researchers. but they are all means the same. IDIC 15, inverted duplication 15, extra markers inverted duplication 15, partial tetrasomy 15. Jacobson syndrome, also called as a terminal 11q deletion disorder. This is a very rare disorder. Those affected have normal intelligence or mild developmental disability with a poor expressive language skills. Most have the bleeding disorder called Paris Troussia syndrome, the Klinefelter syndrome, XXY. Men with Klinefelter syndrome are usually sterile and tends to have a longer arm and legs and to be taller than their peers. The boys with syndromes are often shy and quite have a higher incidence of speech delay and dyslexia. During puberty, without testosterone treatment, some of them may develop gynecomastia, Patwa syndrome, also called as a D syndrome or trisomy 13. The symptoms are somewhat similar to those of trisomy 18, but they do not have the characteristic hand shape. The small supernumerary marker chromosomes. This means there is an extra abnormal chromosomes. The future depends on the origin of extra genetic materials. The cat-eye syndromes and isodendritic syndromes 15 syndrome are both caused by the supernumeracy marker chromosomes, as in Pallister-Killian syndrome. The triple X syndrome, triple X, the triple X girls tends to have tall and thin. They have a higher incidence of dyslexia. The Turner syndrome X instead of XX or XY. In Turner syndrome, the female sexual characteristics are present but underdeveloped. The people with the Turner syndrome often have a short stature, a low hairline, abnormal eye features, and bone development, and a caved-in appearance to the chest. XYY syndrome. The XYY boy are usually taller than their siblings, like XXY boys and XXX girls. They are somewhat more likely to have learning difficulties. The wolf Hirschhorn syndrome, which is caused by the partial deletion of short arm of chromosome 4. It is characterized by severe growth retardation and severe to profound mental health issues. The Jane chromosomes, these are the chromosomes which are extremely large compared to the normal chromosomes. Such chromosomes called Jane chromosomes occurs in some animal cells. Two types of Jane chromosomes are known. One, lamb brush chromosomes. Two, polytene chromosomes. The lamb brush chromosomes, it was discovered by Ruckert. Lamb brush chromosomes were seen in oocytes of several different animals like shark, amphibians, reptiles, and birds, most distinct in a diploid stage. 3. The change size of these chromosomes due to the increase in size of chromonema, not due to the number. The chromonema grows to form lateral loops. These loops gives it a brush-like appearance. That is why these are called lamb brush chromosomes. 4. Lamb brush, the chromosomes concerned with the vitellogenesis, yolk formation. And 5. Synthesis of mRNA occurs on the lateral lobe. Note the compound chromosomes, a genetic engineering procedure which produces two chromosomes, in one of which the left arm of two original chromosomes joined together and the two original right arms are joined together, used in the genetic control of insect populations. Homologous, the chromosomes of a matching pair in the diploid complement that contains alleles of a specific genes. 2. Ring chromosomes, a chromosomes in which both ends have been lost, deletions, and the two broken ends have reunited to form a ring-shaped figure. 3. Z chromosomes, the sex chromosome in animals such as poultry, in which the female is heterogametic sex. The male has the ZZ genotype and the female The ZW genotype, polythene chromosomes. It was first observed by E.G. Balmiani in Chromosomes. These are commonly salivary gland chromosomes. They are found in this larvae of certain dipterans. Such banded chromosomes occurs in the larval salivary glands, midgut epithelium, and rectum, and malphigian tubules of various genera, Drosophila and Sierra. For the polythene chromosomes get their name from the fact that they are formed by many parallel chromatids, often more than the thousand strand, which do not separate from one another following the duplication. Along each chromatid strand, some regions of the chromatin are tightly coiled and other regions are less coiled, with the results that the polythene chromosomes appears to consist of light and dark bands when observed under the microscope. 5. The main functions of polythene chromosomes is to carry genes which ultimately controls the physiology of an organism. These genes are formed of DNA molecules. The supernumerary chromosomes in nuclei of some plants and animals, in addition to normal chromosomes, are present one or more accessory or supernumerary chromosomes. The Wilson first discovered these in 1905 in Hemipteran insect Metapodius. Since then, they have been found in variety of insects and in many higher plants. In Metapodian, these are derived from Y chromosomes. More commonly, their ancestry is entirely unknown. Generally, they are of smaller size and appears to be genetically inert. This table gives the total number of chromosomes, including sex chromosomes, in a cell nuclei. For example, the human cells are diploid and have 22 different types of autosomes. Each present has two copies and two sex chromosomes. This gives 46 chromosomes in total. The other organisms have more than two copies of their chromosomes, such as breed white, which is hexaploidy and has six copies of seven different chromosomes, 42 chromosomes in total. Chromosomal numbers in some plants, plant species, Arabidopsis thylana diploid 10, uh, diploid 14, maize diploid or paleotetraploid 20, young corn wheat diploid 14, durum wheat tetraploid 28, bread wheat hexaploid 42, cultivated tobacco tetraploid 48, adder's tongue fern diploid. The chromosomal numbers 2n in some animals species common, fruit fly 8, guppy Polycilla reticulata 46, earthworm Octodrillus complantus for 36, domestic cat 38, laboratory mouse 40, rabbit Oricatolagus caniculus 44, hares 48, gorillas chimpanzee 48, elephant 56, donkey 62, dog 78, guinea pig 64, garden snail 54, Tibian fox 36, domestic pig 38, laboratory rat 42, Syrian hamster 44, human 46, domestic sheep 54, cows 60, horse 64, kingfisher 132. The common chromosomal number in other organisms, Trypanosoma brucei, larger chromosomes 11, intermediate chromosome 6, and microchromosomes approximately 100. The domestic pigeon, Columba divia domesticus, larger chromosome 18, intermediate nil, and microchromosome 59 to 63. The species chicken, larger chromosome 8, intermediate chromosome 2, sex chromosome, and microchromosome 60. Transposon 3.3. It discovered by Barbara McClintock. The genome evolved by rearranging the existing sequence. The rearrangements are sponsored by processes internal to the genome. There are two of major causes of rearrangements. One, unequal recombination results from mispairing by cellular system of homologous recombination. Two, another major causes of variation is provided by the transposable elements or transposons. These are discrete sequences in genome that are mobile. They can transport themselves to other locations within the genome. The transposons that mobilize via DNA are found in both prokaryotes and eukaryotes. Each bacterial transposons carries genes that codes for enzyme activities required for its own transposition. In eukaryotes, these enzymatic functions are not well characterized. A genome may contain both functional and non-functional elements. Many elements in a eukaryotic genome are defective and have lost the ability to transpose independently, although they may still be recognized as a substrate for transposition by enzymes produced by the functional transposons. A eukaryotic genome contains a large number and varieties of transposons. Insertion sequence simplest transposons. The simplest and shortest transposon known as insertion sequence were first found in bacteria. They are designated IS1, IS2, etc. The typical insertion sequence are 750 to 1500 base pairs with the terminal inverted repeats of 10 to 40 base pairs. The insertion sequence only encodes a single enzyme that transposes the enzyme needed for the movement. The inverted repeats is a region that contains two open reading frames. This gene product is a transcriptional regulator that controls the production of transposase and of itself. An insertion sequence is a transposition that encodes for genes needed for transposition flanked by a short inverted terminal repeats. The target site at which the transposons is inserted is duplicated during the insertion processes to form two repeats in direct orientation at the ends of transposons. The length of a direct repeat is 5 to 9 base pair and is characterized by characteristics for any transposon. The transposon has a inverted terminal repeats and generated direct repeats of flanked in DNA at the terminal site. The isDNA is always flanked by very short direct repeats. The sequence of direct repeat varies among individuals transposition events undertaken by the transposons, but the length is constant for any IS elements. The most common length for the direct repeat is 9 base pair. The table inverted repeats defines the end of transposon. IS1 9 base pair, 23 base pair, 768 base pair, target section random. IS2 5 base pair, 41 base pair, 1327 base pair hot spots. IS4 11 to 13 base pair, 18 base pair, 1428 base pair, AAA N20 TTT. IS5 4 base pair, 16 base pair, 1195 base pair hotspots. IS10 or 9 base pair, 22 base pair, 1329 base pair, NGCT IS50 or 9 base pair, 9 base pair, 1531 base pair hotspot. IS903 9 base pair, 18 base pair, 1057 base pair random. The composite transposons. These transposons are named Tn followed by the number. One class of larger transposons are called composite elements because a central region carrying the drug marker S is flanked on either side by arm and consists of IS elements. Some common transposons, example, TN903, TN10. Left end IS903, IS10LB non-functional. Markers KAN resistant. Right end both ISNs functional, IS10 are functional. Example TN5. Left end IS50L non-functional. Markers tetracycline resistant, kanamycin resistant, and right end IS50 are functional. The transposons can carry other genes in addition to those coding for transposition. The transposons have a central region flanked by an IS element at each end. Either one or both IS element of the composite transposition may be able to undertake the transposition. A composite transposition may be transposed as a unit, but an active IS element at either end may also transpose independently. The structure of a composite transposon. ISL left, ISR right, transposon marker. IS module has inverted repeats. IS module has inverted repeats. A major force supporting the transposition of composition. Transposition is a selection of for a markers carried in the central region. An IS10 molecules module An IS10 module is free to move around on its own and mobilize an order of magnitude more frequently than TN10. The TN10 is held together by selection for tetracycline, so that under selective conditions, the relative frequency of intact TN10 transposition is much increased. A. Composite transposition, and B. Structure of feature of composite, and B. Simple transposition. Transposition, replicative, and non-replicative mechanisms. In replicative transposition, the element is duplicated during the reactions, so that the transposing entity is a copy of the original elements. The replicative transposition involves two types of enzymatic activity: the transposase that acts on the ends of original transposon, and resolvase that acts on the duplicated copies. A group of transposons related to TNA moves only by the replicative transposition. The replicative transposition processes through the co-integrated. The basic structure involved in the replicative transposition are follows: transposon and fusion in it forms integrate, and it forms a recombination forms a separate. In non-replicative transposition, the transposing elements moves as a physical entity directly from one side to the another side and is conserved. The insertion sequence and composite transposon TN10, TN5 use the mechanisms. The non-replicative transposition proceeds by breaking and reunion. The conservative transposition describes another sort of non-replicative events in which the element is excised from the donor site and inserted into the target site by a series of events in which every nucleated bond is conserved. Transposons causes rearrangement. The transposon causes rearrangement of DNA. The homologous recombination between the multiple copies of transposons causes a rearrangement of host DNA. The homologous recombination between the repeats of a transposon may leads to precise or impairs excision. TNA transposition organization. The transposition of a TN10 has multiple controls. The multiple copy inhibition phenomena of multiple copy inhibition reveals that the expression of the IS10 transposes gene is regulated. It reduces the rate of transposition of any one copy of a transposon, which other copies of same transposons are introduced into the genome. The multiple mechanism affects the rate of a transposition. The replicative transposition of a TNA requires a transposase to form the co-integrated structure and resolvase to resist the two replicons. The best characterized transposons in plants are controlling elements of maize, which falls into several families. Each family contains a single type of autonomous elements analogous to the bacterial transposon in its ability to mobilize. The first transposon was discovered in the plant maize, Zea maize corn species, and it is named as a dissociator DS. The transposition of maize element AC is non-replicative, probably requires only a single transposase enzyme coded by the element. The P element in Drosophila melanogaster are responsible for hybrid dysgenesis, which could be a 4 hour of speciation. Eukaryotic transposable elements. Eukaryotic transposable elements falls into one of the two classes called class 1 and class 2 elements. The class 1 elements are also called as RNA elements because the DNA elements in the genome is transcribed into an RNA copies. The RNA copies is converted back into the DNA copies that can then inserted into another target sites in the host genome. The elements are also called as retro elements because their movements called retrotransposition is characterized by the reverse flow of genetic information from RNA to DNA. There are three main subclasses of retrotransposition. One, viral encoded reverse transcriptase to reverse transcribe RNA to DNA have long terminal repeats LTR similar to retroviruses. Two, lines encodes reverse transcriptase lag LTRs transcribed by RNA polymerase 2. 3. Non-viral superfamily do not code for reverse transcriptase that it's transcribed by RNA polymerase 3. Common type viral superfamily S. cerevisiae, copia D. melanogaster. Lines L1 human, B1, B2, ID, B4 mouse. Normal superfamily signs mammals pseudogenes of pole 3 transcript. Termini, long terminal viral superfamily, long term repeats, and lines no repeats, normal superfamily no repeats. 3. Target repeats viral superfamily 4 to 6 base pair, lines 7 to 21 base pair, and normal superfamily 7 to 21 base pair. 4. Enzyme activities viral superfamily reverse transcriptase and or integrase lines reverse transcriptase or endonucleases. Normal superfamily none or non-coded for transposon products. 5. Organization viral superfamily many may contains interons removed genomic mRNA. Lines 1 or 2 uninterrupted O or F. Normal superfamily no interons. The class 2 elements are called DNA elements because a class 2 elements itself moves from one site in the genome to another. The single transposable elements discovered genetically in the maize were class 2 elements that lead to unusual spotted kernels due to their excision from the gene responsible for pigment production in the kernel. Class 1 class 1 transposable elements and flanking DNA donor DNA. In case of class 1, transcription forms RNA intermediate, reverse transcript forms DNA intermediate, and insertion it forms a target DNA, it forms transposable mobile element. In class 2, the forms with donor it forms excision formal in insertion site donor DNA, it forms a DNA intermediate and forms transposable mobile element. Figure 2 classes of transposable element in eukaryotes. Retrotransposons, the transposable elements that transpose through an RNA intermediate utilizing a reverse transcriptase are termed retrotransposons. They are found primarily in eukaryotic organisms. They can be divided into two types, the LTR and non-LTR transposons, based on the presence or absence of direct repeats at the end of the element called long terminal repeats. The long terminal repeats retrotransposons, viral retrotransposons are similar to retroviral DNA, similar to those of in retroviral DNA. The viral retrotransposons are flanked by 250 to 600 base pair. The long terminal repeats and like retroviruses encodes the reverse transcriptase and integrases. The retroviruses have a single standard RNA genome. In the host cytoplasm, the retroviral genome RNA first gets reverse transcribed into double-stranded DNA and then integrated into the host genome. The double-stranded DNA is inserted into the host genome by transposition, like event that generates a short direct repeats of the target DNA. A retroviral sequences that is integrated into the host genome remains as an endogenous proviruses. The viral retrotransposons moves in the genome by being transcribed into the RNA, which then undergoes reverse transcription and integration into the host cell chromosomes. The viral transposons are abundant in lower eukaryotes such as yeast, example TY elements, the Drosophila example copia elements. Figure the general structure of viral transposons. The central protein coding region is flanked by two long terminal repeats LTR. Like other mobile elements, the integrated retrotransposons have a short target site direct repeats at each end. The protein coding regions constitutes 80% or more of the retrotransposons and encodes reverse transcriptase, integrases, and other retroviral proteins. The first step in the transposition of viral retrotransposon is the synthesis of RNA copies of inserted viral retrotransposons by RNA pol 2. This transcript encodes a reverse transcriptase enzyme that makes a double-stranded DNA copies of RNA molecules via an RNA DNA hybrid intermediate, precisely mimicking the early stage of infections by retroviruses. The analogy continues as a linear DNA molecules uses an integrase to integrate into the randomly selected site on the chromosome. The non-LTR retrotransposons, the non-viral retrotransposons lacks LTR and have an A bar T rich stretch at one end. The non-viral retrotransposons are the most frequently encountered transposons in mammals. The most abundant non-viral transposons in vertebrate are called signs. A short interspersed linear element, nuclear element, a short interspersed nuclear element, and lines, a long interspersed nuclear elements. The lines are autonomous and retrotranspositionally active elements. The most common lines in human genome constitutes the line 1 L1 line family. The L1 elements are approximately 6 kb in length and in most species highly accumulated in the sex chromosome compared to the autosomes. Some 5 lakh copies of L1 elements occurs in the human genome, accounting for about 17% of total human genome. The L1 element usually are flanked by a short direct repeats, the hallmark of mobile elements. The consensus sequence containing two long open reading frames, the ORFI, the the open reading frame 1 encodes an RNA binding proteins, and the open reading frame 2 ORF2 encodes the protein that has both endonuclease and reverse transcriptase activity. The figure, the genetic organization of a typical line, the shorter open reading frame ORF1 about 1 kb in length encodes an RNA binding protein. The longer open reading frame 2 about 4 kb in length encodes a bifunctional protein with a reverse transcriptional and DNA endonuclease activity. The signs are the SINES are 100 to 400 base pair long non-autonomous sequences, and thus requires the enzymatic machineries of line elements for retrotransposition. The signs or transcribed by RNA polymerase 3. The most common signs in human genome is ALU elements. ALU elements frequently contains a site of restriction enzymes ALU and consequently are called ALU elements. These about 300 base per sequence are scattered throughout the human genome constituting approximately 10% of the total DNA. A related transposable elements B1 is found in mouse genome. Both transposable DNA sequences are though to have evolved from the gene for 7SL scRNA and a non-coding RNA involved in the movement of a polypeptide in the organelles. The comparison between the viral and non-viral retrotransposons, enzymes, viral retrotransposons, reverse transcriptase and or integrase, non-viral transposons, reverse transcriptase and endonuclease. The LTR, the LTR, viral retrotransposons present and non-viral retrotransposons LTR absent. Target repeats viral retrotransposons 4 to 6 base pair and non-viral retrotransposons 7 to 21 base pairs. The common type TY and COPIA viral retrotransposons and non-viral retrotransposons L1 and B1. Viral transposons. The E. coli bacteriophage MU is an unusual phage. It is a temperate phage. It has both lytic and lysogenic growth cycles. When DNA of a phage MU enters E. coli, its host, it integrates into the host genome. However, it does do so by a site-specific recombinations. Rather, it integrates randomly into the host genome by a transpositional mechanism. When MU replicates, it does so by replicative transposition, even in which each copies of MU is inserted somewhere else in the genome. Unlike other viruses, the DNA of MU is never found replicating as an independent molecules free of the chromosomes. The segment of DNA packed into the virus particles contains a whole MU genome plus small stretch of host DNA at the ends. Thus, the whole of MU genome is a transposon that happens to have gene that packets DNA as well as genes that regulate its transposition.